So how did I get into this? So this actually started because Paul asked me when I did a chief residency during my fellowship. So we had a sort of brief trial at Mount Sinai of the Duke method of having a couple of more senior chiefs. And so I was one of those folks. And so during my nephrology fellowship, Paul said, would you write this book chapter with me on this disease HIVAN that I barely knew anything about at the time? So I, in the best office I ever had during my career, the chief's office at the VA in the Bronx, this enormous office with a great big long couch for residents to sit on and a little candy jar that I think I ate most of the candy from, not the residents. But I sat there and I had spread out along the couch so that no one could sit on it, all of these articles printed out, because that's what you did back then, about HIVAN. So by the time I'd finished this article, I felt like I knew not everything, but probably as much as anybody knew about sort of the full spectrum of disease. So I had learned about primate models of HIVAN and murine models of HIVAN and what we knew about the epidemiology of HIVAN, and put it all into this book chapter that went into a book, Genetic Diseases of the Kidney, that should have been the sort of ultimate textbook on this topic. These, for a nephrology audience, would recognize all four of these editors are really sort of prominent leaders in the field of nephrology or genetics. Rick Lifton is a geneticist at Yale. The others are nephrologists. And this really should have been sort of the end all be all. And it's still a really useful reference for monogenic diseases, sort of pediatric processes that are classic genetic diseases. But unfortunately, the year that this book was published was the year that chromosome 22 was identified as the major source of genetic susceptibility among African Americans to kidney diseases, including HIVAN. So the initial studies identified a gene called MyH9, which is now known not to be the primary culprit. That was picked as a candidate gene out of a big locus on chromosome 22 because it was known to be associated with the Mendelian disease, one of the diseases that's still accurately described in this book. And then a couple of years later, it became clear that the actual gene is this gene APOL1 that is known to be a really strong predictor of HIV-associated nephropathy. So despite this, despite the fact that this book pretty much just sits on my office bookshelf for decoration now and probably is rarely used, certainly for in any of the complex genetic kidney diseases, it really did kind of launch my career in HIV and kidney disease. We initially were studying, and you can see that was the original title of the Klotman's P01 was pathogenesis of HIV-associated nephropathy, and that was really the primary area of research in their lab at the time. For people who don't think about kidney disease very often, HIV-associated nephropathy was the classic kidney disease, and remains, I guess, the classic kidney disease of HIV. Originally described in 1983, so just a couple of years after the first cases of AIDS were described. And at that time, and to this day, was a disease that was seen almost exclusively in individuals who were of African descent. So these were largely African Americans, in some cases that were seen in Haitian immigrants. It presented with a really very specific kidney pathology. It's a collapsing form of focal segmental glomerulosclerosis. Which is, again, nephrologists and renal pathologists tend to be very descriptive in their descriptions of diseases. And again, this was, collapsing. It's collapsed because things are kind of pulled in on themselves. It's focal because it doesn't affect everything in the kidney, and it's sclerosis because it's scarring. There are also these really dramatic tubular changes and tubular and interstitial damage. And this is really the first kidney disease that was linked directly to HIV infection. It typically occurs in patients with very advanced HIV or AIDS, at least in its classic form. We now know that genetic susceptibility to this disease is linked strongly to APOL1. So this has been used as a model disease. So in addition to being an important disease clinically, particularly early in the HIV epidemic, but even today among patients who are either undiagnosed and not yet on ART, or patients who are non-adherent, we still do see HIVAN not so infrequently. But I think now probably its major importance has been as a model disease. So there's a really great mouse model. The classic mouse model is an HIV transgenic mouse called TG26, which expresses a GAG-Pol deleted HIV transcript, and has been used to really describe, probably we know as much or not, if not more, about the pathogenesis of HIVAN as any other kidney disease because of the availability of this mouse model. We also have learned a lot about common kidney injury pathways from this model, so it's been useful beyond HIV-related disease. And human HIVAN, the mouse does not express APOL1, but human HIVAN is really the strongest model of APOL1 mediated kidney disease. And so a lot of the geneticists have actually been very interested in HIVAN to help understand actually the mechanisms and also a little bit more about the relationship between APOL1 and HIVAN. So essentially variants in this gene APOL1 explain a lot of the increased risk of chronic kidney disease in African Americans. Those data were first published. Again, all of these studies included some patients with HIVAN, but also non-HIV related FSGS, that same sort of similar looking pathology. And really suggested that although this doesn't explain everything, that this explains a lot of the reason why we see such a burden of kidney disease in patients who are African American. This association with other kidney diseases is sort of on the 4 to 20-fold increase in risk among people who have the high-risk genotype of APOL1. But it's up to, actually there's one study with 100-fold, but I'd say closer to 90-fold increase in the odds of HIVAN in some populations. So again, this is a really, really strong association with HIV-related kidney disease. There are 2 missense variants of APOL1, which are called G1 and G2, that are found only on African chromosomes, and individuals who are homozygous or compound heterozygotes for these genes or for these alleles have a much higher risk of chronic kidney disease. That includes non-diabetic kidney disease, FSGS, and HIVAN. There's also been associated with an increased odds of proteinuria in HIV positive women. This was a study that I was involved in with Michelle Estrella from the WISE cohort. And these were HIV positive women, both with stable HIV and with advanced disease or history of AIDS. And the relationship between APOL1 and proteinuria was actually the highest among women who didn't have a history of AIDS. So these were women who had stable asymptomatic HIV infection. And so suggesting that it's not all HIVAN, but that there's also some association with proteinuria that's beyond that. This is important because kidney disease in general, but even proteinuria, so even isolated proteinuria has been associated with adverse outcomes in the HIV population and in the general population. So increased risk of cardiovascular events and death in people who have proteinuria. How and why these variants in APOL1 are associated with kidney disease remains controversial. There are a lot of different theories, and I think beyond the scope of this talk, what they are. And the field tends to gets a little heated when some people don't really get along with each other in this area. So I've tried to stick to the epidemiology in APOL1 because the mechanism folks are getting they get a little cantankerous now and again. Interestingly, this is a story that's sort of reminiscent of sickle cell and malaria. The heterozygous state, so either G0, the wild type, G0, G1, or G0, G2, is actually protective against African trypanosomes. So both G1 and G2 variants either decrease or completely eliminate binding to the serum resistance associated protein and result in trypanolysis. These variants also probably, or APOL1 probably has other functions in innate immunity. And there's actually been studies to suggest that it may restrict HIV replication in vitro. But these are data from Sherry Winkler's group. Sherry Winkler is one of the geneticists who's been most interested in this. Did not find any clear effect on HIV risk of infection or severity of disease in humans, suggesting that although that's one of the sort of hypothesized reasons for the really strong association with HIVAN, it doesn't, at least so far we haven't found any evidence that that's the case. Interestingly, there's also some controversy over the possible increase in kidney disease risk with a single risk allele. So, so in these heterozygotes, is there possibly an increased risk? Most geneticists consider this to be a recessive model, meaning you have to have both, you have to have two copies of a variant of a non-wild type allele in order to get it. But there are certainly some data to suggest that there might be some additive effect, meaning that even a single risk allele might put you at increased risk. And some of the strongest data are actually coming from the HIV population, largely unpublished data. But for example, we looked at a group of HIV positive individuals with FSGS who did not have classic HIVAN, but just sort of a regular looking FSGS. They were all more stable antiretroviral treated patients. And almost none of them had two risk alleles for APOL1, but the vast majority of them had a single risk allele. So certainly there's some people who would consider that maybe HIV is a strong enough second hit that even having one risk allele is enough. So that's an area of ongoing research in our group and others. What about, so I mentioned that HIVAN is really a disease of advanced HIV infection or AIDS. What about kidney disease in patients with early HIV or stable HIV, which is more of what we see these days, hopefully, although we unfortunately see enough patients with more advanced disease. These are data from the START trial. I think probably most people here are familiar with START, but for anybody who's not, START randomized over 4,600 antiretroviral naive adults who had a high CD4, so a CD4 of over 500, to immediate versus deferred ART. So basically they either started right away or they waited until their CD4 was less than 350 or they had a clinical indication to start. And the study was stopped early, and actually between the beginning of the study and when it was terminated early, the Department of Health and Human Services guidelines began to recommend immediate ART anyway. But the study was stopped early because there was a clear benefit to immediate ART in terms of the primary endpoint, which was serious AIDS or non-AIDS events with a really substantial reduction in risk. But what about kidney disease in this population? So we looked at baseline. It turns out that there was probably a little bit more kidney disease, in particular proteinuria, than would be expected for this young population. So the median age was in the 30s. And it looked more like the Framingham population, which is kind of a bunch of old white folks in the heartland. So it was, again, probably a little bit more kidney disease than you would expect in these individuals who were ART naive and had CD4s above 500. And we saw a benefit of immediate ART on the GFR slope. eGFR stands for estimated glomerular filtration rate for the non-clinicians in the audience. And this is just a marker of kidney function. And so essentially we saw a benefit. So you started ART right away and your GFR was declined at a slower rate than those who started later. Interestingly, there was a really strong interaction with race. And this was something that we thought about a priori. And when we looked at this benefit was seen only in individuals who are of Black race, self-reported Black race. So there was no difference in the GFR slopes in individuals who were self-identified as white or other race, but among Blacks, this difference was even larger. So that suggested to us maybe this was an APOL1 effect, and maybe we were treating early HIVAN. So these individuals with high CD4 certainly weren't going to present with the full-fledged collapsing FSGS and super sick, but maybe they just had some a little bit of proteinuria, and this was going to be an APOL1 effect. And we were actually surprised. We haven't finished analyzing these data yet, but when we genotyped, about 50% of the START cohort consented to a genomic substudy, and they consented to that prior to randomization. So randomization is protected in that study, meaning that there were about the same number of individuals in both arms, for example. And so we genotyped these individuals for APOL1, and you can see that overall, less than 2% of the overall population had the high-risk genotype, meaning 2 of the risk alleles. Less than 8% of the Blacks had 2 risk alleles. And so this probably doesn't explain the effect that we're seeing. But interestingly, almost 40% of the Black participants have at least 1 risk allele. So we're in the process now of analyzing these data to see if that sort of HIV is a second hit phenomenon might be holding true in this population and perhaps having a single risk allele is enough to increase or to make you prone to HIV having negative effects on the kidney, where antiretroviral therapy would be expected to have a benefit. So I mentioned that HIVAN is a disease of early or untreated, or advanced or untreated HIV infection. So we should be seeing less of it. These are data from a collaboration with the pathologist from the HIVAN program project grant, Vivette Degati, who's up at Columbia, and they have a huge referral population for biopsies. And so they look at biopsies from all over sort of the tri-state area and beyond. And they compiled about 460 biopsies that were done in HIV positive individuals between 2010 and 2018. And you can see that actually HIVAN was still the third most common diagnosis. Now this is going to be probably overrepresented here because HIVAN presents with a lot of proteinuria and things that make nephrologists want to do biopsies. So this is probably still more than, this doesn't reflect the percentage of HIVAN in all the patients, all comers with kidney disease, but it does suggest that we're still seeing, a decent amount of HIVAN. The two most common diagnoses were actually immune complex glomerulonephritis and diabetic kidney disease, which I'll come back to talk about in a minute, as well as antiretroviral toxicity, in particular tenofovir. And I'll note down here also a bunch of FSGS that was not HIVAN. So it didn't have some of the features of HIVAN like collapse or some of those tubular interstitial changes, but still considered an FSGS. And there's some work going on in this area. And I will not talk about acute kidney injury, but this does remind me just to say that probably a lot of the chronic kidney disease that we see in patients with HIV, particularly those who were sicker earlier in their course of disease, is the result of repeated episodes of acute kidney injury, which we know can lead to chronic kidney disease. I wanted to just have one slide here on immune complex kidney disease. I keep hoping that I'll find somebody who wants to collaborate in this area because it's a really understudied area of kidney disease in HIV. And as you can see, it's actually not uncommon. It's the most common cause of biopsy-proven kidney disease in HIV. So unfortunately, this is a wastebasket of really heterogeneous kidney diseases, everything from IgA nephropathy to lupus nephritis to hepatitis B and C related glomerulonephropathies. And that's made it really hard to study. So we don't know much about whether it's related causally to HIV infection, unlike HIVAN, what the natural history of this disease looks like, whether antiretroviral therapy makes a difference. So this is partly because of this heterogeneous spectrum of disease, and unfortunately, partly because of the heterogeneous spectrum of disease, there's no animal model to study this in. So unlike the TG26 model for HIVAN, there's really not been as easy a way to study this. So the little bit of data have been fairly tedious data from looting antigen antibody complexes, immune complexes from individuals with these diseases and looking for HIV antigens, for example. So it's been fairly tedious work to suggest that at least in a small subgroup of these cases, there probably is a direct effect of HIV infection. Many of the others, some of these lupus-like ones, for example, may have an association with HIV that is sort of more related to the systemic immune dysregulation, but not direct effect of HIV. So this is, again, an area for future study. I think it's an important area. I think at least our former program officer at the NIDDK would be super excited if we did something because he's done a little work in this area, but it's just it's been a hard area to study. You saw that diabetic kidney disease was the second most common cause of biopsy-proven kidney disease in patients with HIV in sort of the modern era. And interestingly, comorbid kidney disease is the vast majority of what I saw. So when I was the embedded nephrologist in the HIV clinic at Mount Sinai, probably 50% or more of the patients that I saw were diabetic nephropathy or hypertensive kidney disease or some combination. And so we actually at one point went back and looked. These are data from the veterans, from the VAX cohort, the Veterans Aging Cohort Study, which is essentially an EMR cohort of HIV positive individuals and age and site matched HIV negative individuals, and looked at individuals who had preserved GFR at baseline and looked at progression to GFR less than 45, which we would consider stage 3B chronic kidney disease, sort of the first I think the first GFR-based cutoff that people really always consistently considered to be a disease and not just a sort of fluke of our GFR equations. And you can see that the risk was lowest in individuals with neither HIV nor diabetes and highest in those with both. The statistical test for synergy, for sort of, I mean, it's not a specific, but anyway, we couldn't prove that there was synergy here, but there's definitely at least an additive effect of diabetes and hypertension, diabetes and HIV. And in an animal model, in the murine model, if you induce diabetes in a black 6 mouse, black 6 mice are pretty resistant to kidney disease, both diabetic and kidney disease and HIVAN. But if you have a transgenic, a TG26 or HIV transgenic black 6 mouse with the diabetes induced, you actually do get kidney disease. So suggesting that there is, again, some at least additive effect in the animal model as well. And in that model, there was significant upregulation of inflammatory pathways, which would be one potential mechanism for this association. I have to say I was relieved by these data because I always felt like all of my diabetics in the HIV clinic went on to dialysis, and I felt like maybe it was just me, but I think there's actually something about the disease itself. The other thing that was there, and I'm not going to spend a huge amount of time on this slide, but I did just want to not just pick on tenofovir. These are relatively old, like sort of oldie but goodie data from a paper from the EuroCETA, an HIV, European HIV cohort that were published in 2010. And essentially they looked at cumulative exposure to every antiretroviral therapy individual agent that was available at the time, and the risk of a confirmed creatinine clearance less than 60. And indinavir, nobody was surprised to see indinavir, and really probably nobody was surprised by then to see tenofovir was associated with an increased risk of decreased kidney function. There's a lot of furor about atazanavir, and maybe a little bit less about lopinavir, ritonavir, because people were using it less by then. I'll just point out, so I'm not really going to spend a lot of time talking about the protease inhibitors, but as a class, they have been sort of reproducibly associated with decreased GFR, and there's some controversy about whether that's because they're affecting creatinine secretion directly, whether it's all just a toxic, direct nephrotoxic effect, like with indinavir, which was associated with kidney stones and crystals, which cause inflammation. But in any case, essentially all of them, with the exception of darunavir, and darunavir's really sort of stood out as the one that's reproducibly not associated with decreased GFR. I'm going to spend a little bit more time talking about tenofovir just because that's been an area of active research for me. Tenofovir, at least in some individuals, can cause a direct tubular injury. These are proximal tubules that none of which look normal enough for, this is probably the most normal looking tubule here where the cells are close together. But all of these, there's marked atrophy, cell death and loss of this nice little, they're supposed to have a little furry brush border on them that's all lost. So these are tubules that are damaged. There's a ton of mitochondrial injury. And so the question is, does this is sort of a rare event to see this really flamboyant, really overt injury to the tubules, but is tenofovir doing something more broadly in a larger population? And one thing one place that we saw a suggestion of this is actually back in the START study. So we truncated our initial analysis right here around 3 years, and that was because we had pretty sparse data, follow-up data beyond 3 years. But we did notice that this was already starting to happen, and this is actually borne out in longer-term follow-up. That the curves converge and then cross, suggesting that eventually being on antiretroviral therapy for longer is associated with worse GFR outcomes. And so in addition to looking back sort of at the early part of this curve, the improvement with initiation of ART, we're looking at some more detailed analyses of ApoL1 and urine albumin. At this point, we're actually interested in change in kidney injury biomarkers and whether there's, whether current antiretroviral therapy is associated with kidney injury in this population. Again, I won't spend a whole lot of time on PrEP, but just because the same drugs are used for pre-exposure prophylaxis, FDC, TDF in particular, approved for pre-exposure prophylaxis. Again, there was really no signal for kidney injury in the randomized trials, but one, I think probably the most important factor here is that adherence was pretty lousy in most of those studies. And not only do you not get benefit, but you also don't get a whole lot of toxicity from drugs that you don't take. These are data from the Partners PrEP study where we saw essentially this kind of looks like early curves from the tenofovir studies in HIV, where we saw a decline in GFR in individuals who were randomized to active PrEP, either TDF or FDC TDF. And there were 2 participants, both in the active PrEP arm, who got to a GFR of less than 60. Both of these were after long-term therapy. This was 30 and 36 months. So this is not short-term PrEP use that was associated with toxicity. And I think that's important to note. They saw pretty similar results in the IPREX study. These are data from Partners PrEP. And these 2 studies, in particular Partners PrEP, had very high adherence to antiretroviral therapy because these were serodiscordant couples. So again, I didn't want to belabor the TDF data because I think everybody's now thinking, well, what about TAF? Because we're sort of abandoning, I think many people are abandoning TDF in favor of the newer prodrug of tenofovir, TAF. The reason for this is, and these are just some data to give some rationale for it, the risk factors for, at least for overt toxicity with TDF, included things that raised the plasma tenofovir concentration. So things like ritonavir boosted protease inhibitors, that would also be true with cobicistat or the hep C protease inhibitors, lower baseline creatinine clearance, female sex, low body weight, all of these things which were associated with high plasma concentrations of tenofovir. TAF, or tenofovir alafenamide, which is an alternative prodrug, has much lower plasma concentrations of tenofovir at its effective doses, so would be thought to have lower potential for kidney and bone toxicity. For the same reason, actually, it has a less favorable effect on lipids, and I think there's not been much done in this area, but this is not because TAF, at least not thought to be because TAF has a bad effect on lipids, but because the higher concentration, has higher plasma concentrations of tenofovir achieved with TDF actually had a favorable effect. TAF is now also approved for PrEP in addition to HIV treatment. And I'll just say there are limited data on hard clinical outcomes, but so far the data would suggest that TAF is going to be safer in most individuals. I think the population where there's still some question about TAF is largely in patients with and at risk for kidney disease. I would say, and this is just my own personal opinion as a clinician, that I would probably not re-challenge somebody with TAF after they developed clear tenofovir toxicity, because this is still tenofovir. And if an individual develops an AKI or a progressive decline in GFR for other reasons and starts to accumulate tenofovir, it's the same drug and they have the same potential for toxicity. So again, I wouldn't re-challenge somebody if I had alternatives, which these days is more likely. The fixed dose combination of elvitegravir, COBI, FDC, and TAF is now approved in hemodialysis populations. And I think that's been kind of a boon for our patients who really before had to take like 3 individual drugs dosed at weird intervals after dialysis and on different days, and who could keep track of that? And I'm always incredibly impressed with those patients and have been a really strong advocate. And I'll talk a little bit about transplant in patients with HIV, but I've always been a really strong advocate of our patients, because if they could keep up with that weird antiretroviral regimen, then they were going to take their immunosuppressive drugs for the transplant. So, so this has been great for individuals to be able to take just a daily fixed dose combination. But there are some concerns there. And I think some things just to be aware of and think about if for the clinicians in the audience, that the plasma concentrations with TAF in this setting will really look pretty similar to a person with normal kidney function who's taking full dose TDF. So there are some concerns about, particularly in individuals who have residual kidney function, which is associated with better outcomes. So you're on dialysis, but your kidneys still do a little bit of the work. That means you get better clearance overall. And that is associated with better outcomes. There may also be some impact on bone health, and patients who are on dialysis already have plenty of problems with bones already because of parathyroids and high phosphorus and crazy calcium levels. So I think these are important considerations to think about And I also worry in the peri-transplant setting about dosing and drug-drug interactions. So it's just particularly with the COVID. So I said I would talk a little bit about transplant. I'm going to kind of breeze through this as well, because this is not my primary area of focus, but I do think it's a really important area to think about. This is Elmi Mueller. Elmi Mueller actually started kind of a cooler thing. So I'm going to just say kidney transplant in individuals with HIV, I'll back up, I'll come to this first, has been associated with good outcomes, really pretty similar rate of opportunistic infections to the general transplant population. There is a higher rate of acute rejection that could be related to immune dysregulation in HIV, but seems more likely to be at least largely the result of drug-drug interactions, and in particular with the protease inhibitors and cobicistat and the calcineurin inhibitors, which are really the mainstay of immunosuppressive therapy. But I think what's new and interesting is the use of HIV positive donors and HIV positive recipients. And that was something that was really championed and pioneered by this woman. And I always include this because I think she looks like a badass in this picture. My daughter's reading a series of books that are like one-page stories about women, girl rebels, including there was like a woman who was a pirate, and and everything from that to Angela Merkel. And I really feel like Elmi Mueller should be in that book. We're reading the second book, and if she's not in it, I'm gonna write to the author and suggest that they add her. She was a fairly junior transplant surgeon in Cape Town, and she was really devastated by the numbers of potential like healthy looking kidneys that she was throwing in the garbage can because of the high rates of HIV there. So it would turn out to be an HIV positive donor. They'd chunk these kidneys in the garbage can. And at the same time, she knew that patients were dying from end-stage renal disease there because access to dialysis and transplant were really quite limited in individuals, in anybody, but certainly in individuals with HIV. So she decided, I think with the blessing of her institutional review ethics committee, but not her department chair, to start giving some of these organs, HIV positive organs, to consenting HIV positive recipients who would otherwise have died from their disease. And she really found that these individuals did amazingly well. So she's published in 2010 her short-term outcomes, in 2015 longer-term outcomes, showing really these individuals did great. So far, they hadn't had issues with HIV superinfection or new strain, new resistance, and their transplant outcomes were really good as well. And this has changed, led in 2013 to a change in US law and the sort of transplant network policies to allow the use of HIV positive donors. And I like to see him. I'm less excited to see him now that I don't have to look at that other guy on TV 17 times a day, but still makes me happy to see Obama now and again. And that led to the Hope in Action study. There are a handful of centers that I think are doing HIV positive to HIV positive transplants, not directly within this study, but they're all doing it as part of a research protocol. But this is really a large multi-center study that is following graft outcomes, HIV outcomes, and looking at the impact on waiting time in HIV positive recipients of HIV positive donors. Interestingly, I think one sort of unexpected silver lining is that a number of false positive organs have been saved from discard. So these were organs that on screening were felt to be HIV positive, would have just been thrown away in the past, were donated to HIV positive recipients, and then actually on further testing, these turned out to be perfectly, disease-free organs. And so that's been kind of a neat side effect of this. And it's also offered some really neat opportunities to study viral transmission and viral reservoirs. And I am not going to talk about this because I'm sure I can't remember if we've heard recently from Maria or if we should hear soon from Maria Blasi in Mary Klotman's lab and now her own lab about the ongoing work in looking at the kidney as a viral reservoir. I will also just add on a personal note that this has really been incredible for some of our patients, both those with end-stage kidney disease and also for those who would be interested in donating and had never been considered as potential donors before. So there have been altruistic donors have come forward from the HIV community, really excited to donate. And And I know a number of my own dialysis patients were the beneficiaries of this policy, and it's really made a huge impact, I think, for our patient population. In fact, one of my patients asked me, she ended up getting a kidney through the HOPE Act, and she originally, when she was first listed for transplant, asked me about getting a hepatitis C organ, and it was before we were doing that. So she was ahead of her time. Now again, giving hepatitis C positive organs to just about anybody has become the norm, at least anybody who we think is going to be able to take the therapy after. So with that, I just wanted to spend my last couple of slides talking about COVID. It is, after all, 2021, and what talk is complete without at least a slide on COVID. And I just wanted to mention the fact that I think there's a lot of discussion about whether COVAN or COVID-associated nephropathy is kind of the HIVAN of 2020. It really looks just like HIVAN. It's a collapsing FSGS that looks, if you looked at it under the microscope, you wouldn't know whether this patient was HIV positive, parvovirus B19 positive, or COVID positive. But it really looks just the same. And similar to HIVAN, it is strongly linked to the APOL1 risk genotype. It appears to be induced by interferon, which is also the case for HIV. And I think this offers some neat new opportunities for collaboration, some of which we've already started, and some of which I'd still be open to considering. More generally, acute kidney injury is also common in this population. It appears to be multifactorial, so some of it is just sepsis related, some of it is maybe increased clotting, inflammatory cytokine storm, for example. One other area that I think would be important to know more about is that we know that AKI is a strong predictor of CKD in other populations, and there are really no data on what impact this will have on long-term kidney health in patients who are recovering from COVID. So I think this is another interesting area for research going forward. I would like to stop there and leave some time for questions if there are any. Again, a number of people that have been involved in this work over the years, and I just come back to mention both Paul and Mary Klotman, and also Mike Ross, who was a former Duke resident who went on to be a nephrology fellow at Mount Sinai, and actually was by chance my resident when I was a second year medical student. Mike was my second year medical resident, and has really been a mentor and friend to me from that time on, and probably another one of the reasons that I became a nephrologist. But a nephrologist with a distinct interest in HIV. So again, thanks to everybody for your time and attention, and I'd be happy to take any questions.
Thank you. That was a great tour de force, Christina. It looks like we have one question in the chat box about TDF and TAF. Do you see it? Can you see that?
Why can I not see the chat box? Let's see what I can. Oh, here we go. It's the one thing not on my screen. So that's a great question. So the question is, why did TDF and TAF appear to have different toxicity profiles when they have the same active metabolite? And that's actually, that same active metabolite is the reason that I have concerns about re-challenging individuals who've had toxicity with TDF, with TAF, or about giving TAF to individuals who have really very reduced GFR. And the reason is just because people are exposed to less of the active metabolite, less tenofovir, not the diphosphorylated type, but just plain old tenofovir floating around in the plasma. There is less of it, and that is because of the metabolism of TDF and TAF. So TAF is taken up largely as into the target cells as tenofovir, and so there's just less tenofovir in plasma for individuals who are taking TAF, and that's thought to be, it's like a substantial reduction, like 90-fold reduction in concentration. So it really is thought that both the bone toxicity and kidney toxicity are concentration dependent or exposure dependent. So if you give somebody enough TAF, you could give them toxicity too. And that's exactly my reason for concern in people who are at risk for having high exposure to tenofovir with TAF. So largely people with reduced kidney function would be the main people in that category.
Other questions for Christina.
Hi, this is Twile from Infectious Disease. Hi, Christina.
Hi.
Very, very nice talk.
Thank you.
I have two questions, but the first one was more clarification between FSGH and HIVAN.
Sure.
Is it like a spectrum? Does HIVAN sort of the a spectrum? You know, collapsing. You mentioned collapsing FSGH. Can you have HIVAN without FSGH? Yeah. It sounds like you can have FSGS without HIVAN, but is there?
That's a great question. So you can have FSGS without HIVAN, and that would be what that would look like. I kind of came back to this picture just as an example. So there you would have, things would look more like this. Like this is a fairly normal looking tubule, not totally normal, but more normal. This is kind of where the cells are back to back and the tubules are nice and skinny. You wouldn't see any of this. So in an HIV negative or individual, unless they had COVID or part of virus might be 19, you shouldn't see a lot of this, and there's usually not much inflammation in FSGS. It also tends to be less sort of pulled away from, this is the glomerular capsule, and it would tend to be less pulled away. So we can see that in individuals who are HIV positive or HIV negative. And I didn't present those data here, but there is a lot of interest in whether this non-collapsing, sort of non-HIVAN FSGS in an individual with HIV is just true and unrelated, or whether HIV is still a trigger for that disease. And Mike Ross actually has some interesting data comparing HIV positive non-HIVAN FSGS to HIV negative FSGS, and showing that there are some differences there. Suggesting that HIV probably is playing some direct role in those non-classic HIVAN FSGS.
Okay, and thank you. My second question is very curious about the African American susceptibility to HIVAN, and I would imagine the African population in Africa. Do we know anything about the epidemiology of HIVAN there? I mean, working in Asia for 10 years, I know that we hardly see HIVAN in the Southeast Asian population, at least. I don't know about South Asian, but we don't see any HIVAN. And actually, there's some study that actually looks specifically for HIVAN in biopsy, and we don't see HIVAN in Asian population. So I'm just wondering, the African American susceptibility gene, the APO gene, is that seen also in Africa, in Africa?
Yes, it is. So this study here, the one that showed an 89-fold increase in odds of HIVAN with the APOL1 high-risk genotype, was actually from South Africa. There are parts of South Africa where the APOL1 high-risk genotype is quite common, and then other parts of Africa where it's not. East Africa has very low rates of the APOL1 high-risk genotype. It's not uncommon to have a single risk allele, but it's quite uncommon to have two. West Africa, so Nigeria, Cameroon, for example, have been where studies have been done, have much higher rates of the APOL1 high-risk genotype, and that's not so surprising because that's unfortunately the origin of many of the African Americans that were brought to this country as slaves. So West Africans also have a higher rate, and South Africans a higher rate of HIVAN. There's almost no HIVAN in Kenya, for example. We did a biopsy study a number of years ago, and in Kenyans who were ART treated and had either microalbuminuria or overt proteinuria, and we didn't find any HIVAN cases there either. And again, it's pretty consistent with the APOL1 genetic prevalence. So it is the epidemiology of HIVAN really closely follows the genetic epidemiology of APOL1.
Fascinating, thanks.
Yeah.
Other questions? Christina, I'm a little bit curious. What about other viruses that infect the kidney that could be contributing to some of this? I'm thinking about CMV, polyomavirus, et cetera. Any hints that there?
That's a great question.
There may be an issue.
Yeah, so a number of years ago, I gave a talk at one of our ID conferences at Mount Sinai, and one of the infectious disease doctors said, "Well, is there a virus that causes HIVAN?" And I said, "I haven't convinced you it's HIV, but it's but the question of whether other viruses, particularly in somebody who is immunosuppressed, might be contributing to kidney disease, is a good one and has not really been that well studied. So we know that hepatitis B and C co-infection are very common causes of kidney disease in individuals with HIV hep B or C co-infection, and that's actually leading to a lot. I think on that slide with the different biopsy diagnoses, a lot of those immune complex GNs were in fact hepatitis B or C related. But people we have not, and others have not really exhaustively looked at other viruses like CMV, EBV, even some of the sort of the BK viruses, other things that are associated with kidney disease in other settings. And I suspect that there is probably something there. Leslie Brueggemann, who also trained with Paul and Mary, is starting to do some work in that area, both in individuals with HIV and in the general population, looking for other viruses as a cause of kidney disease. So it's I think a really important area of research that certainly not been exhausted, and barely touched.
And one maybe related question. You mentioned in one of the scenarios a fairly profound mitochondrial damage showing up. Again, mitochondrial damage and release of mitochondrial DNA, we are now coming to recognize it's a huge activator of innate immunity. Any thoughts that there could be a cascade here, that somehow you are seeing immune-mediated damage in some of these scenarios?
Sure. So I think the picture that I showed with the horrible looking mitochondria was tenofovir toxicity, which tends to be not very inflammatory. But that may just be because those tubules those cells just die, right? I mean, they just die and slough off, and they don't stick around long enough to exude all their mitochondrial DNA everywhere. You can find a ton of, that's when we look under the microscope in a patient with tubular toxicity of any sort, we see tons and tons of cells there, or dead cells there. So that may just be a question of they're just not there long enough. Podocytes are less likely to drop out. So I think, if other processes like HIVAN causing mitochondrial damage may be more likely to kind of start a vicious cycle like that.
Okay.
Question. And mitochondrial damage is thought to be is one of the many proposed mechanisms for the APOL1 genetic effect.
Okay, good. Any other questions for Christina? All right. Well, hearing none, we again are thrilled that we finally caught up with you, and you're now fully engaged with the Duke CFAR, and we're going to take advantage of that. And we will make sure that we plan this year's retreat around your availability, because we want to make sure that you're there. Thank you so much.
I'll plan my availability around the retreat this year.
Okay, deal.
Great.
All right. Thank you so much, Christina. Thank you, everyone.
Thank you.